at least you don't get lost. The first question is saying that the position of a gene on a chromosome. How do we call the position of a gene on a chromosome? It is called it's called a locus. It's called a what? A locus. That's the position of a gene on a chromosome. Then we go to the next question. They're saying that the diagram below represent the process occurring in the cell. The process, yes. So how do you call this process? If you look at this process, you see that you have a DNA structure, and both strands are acting as a template for formation of the new strand. Therefore, this is DNA replication. I hope you got that answer. So these are 2 marks. Those are 2 marks. Then we go to the next question. They are saying that Jason and Robert are identical twins, meaning that they are coming from the same egg, same ovum. If they're identical twin, which one of the following uses of DNA profile is not possible when considering these identical twins? Number 1, identification of Jason and Robert's father. It looks to be correct. Then they are saying that determining if the woman is Jackson or Robert's aunt. Which one looks to be correct? Also, they're saying it's not. So we are looking for the one which is not used. It could be it could be the function, but it's not used in terms of the identical twins in this case. Distinguishing between Jason and Robert when determining who committed the crime, because they have some differences in the DNA, but most of their DNA is almost the same because they're identical twins. Therefore, this might not work that much, because here you are looking at the differences among them. So this one not really. And then determining Jason or Robert could be, which one could be, or can they be a kidney donor? Yes, this is also correct. So the answer, which looks to be not that much, is this. So gets the 2 marks. Then you go to the next question, is about the graph or the table. They are saying that part of the gene produces a protein that has this triplet TTC, meaning that if you see TTC, it means that it's coming from DNA. Yes. And then you have is being replaced. They're saying that a mutation occurred and then the base triplet became this, meaning C is replaced by T. So it's also still DNA. And then they're saying that the table below gives messenger RNA codon with their corresponding amino acids. So in this case, they're saying the anticodon, they're saying anticodon. Now this is transfer RNA, meaning that I have to convert from DNA to messenger RNA to transfer RNA so that I can get the answer here. The anticodon will ensure the correct amino acid is placed in the normal protein, in the normal this one. So it's gonna be exactly the same, but because amino acid is on there, it's not there, it means that it's gonna be UUU. So it's gonna be it's gonna be this. So you're gonna convert it TC, convert it to, it's gonna be AG, and then convert it from messenger RNA to transfer RNA. So it's gonna be UUC. So that becomes the correct answer. So D becomes the most correct answer. Then we go to the next question, that which amino acid will be found in the protein as a result of the mutation. Yes, which one is going to be correct as a result of the mutation? Here we are seeing that found as a result of the mutation. Since we are looking at the mutation, so we have to go back and look at which one is it. So we have to consider that. So they are saying that which amino acid will be found in a protein as a result of mutation. Amino acid will, sequence will not change or unaffected as okay. Let's find out. When you convert this, because this is messenger RNA, so this is gonna be AAG. So I find out what is AAG. AAG is leucine. No. Yes, it's not leucine, is lysine. And then what about this TTT? When you convert it, it becomes AAA. So what is AAA is the same thing. So the answer here therefore becomes the amino acid will not be changed or affected. So that is the answer. Which of the following describe the event that occur in meiosis but not in mitosis? So in this case, we are looking at what occurs in meiosis but not mitosis. Chromatin network becomes, yes, both they occur. Nuclear membrane disappear, correct. Cytoplasm separate, yes. And then halving. Remember, mitosis keeps the chromosome number. Therefore, here the answer becomes D. So we get that as D. We go to question number 7. Question number 7. What are they saying about question number 7? They're saying that Lindewe suffer from a disorder called hyperhidrosis. This condition is inherited disorder, meaning that you obtained from the parent. Yes, which means that you cannot change it unless you deal with the genes. That result in the person sweating too much. So the scientists have discovered that the dominant allele causes the disorder is homozygous dominant for this disorder. The doctor successfully treats Lindewe's condition by injecting her with the drug to reduce the sweating. Which one of the following statement about Lindewe's treatment with the drug is correct? Treatment with the drug will ensure that Lindewe's offspring do not inherit. You did not change the DNA, so this one is wrong. Affect Lindewe's phenotype. Yes, it could be correct because the sweating is not that much, so you're no longer seeing that sweating. Don't forget that phenotype is what you see. And then they're saying that affect Lindewe's genotype. No, because you're not dealing with a gene. Affect neither Lindewe's genotype. No. So the answer becomes B. So B becomes the most correct answer. Here they're saying that the scientists raise the hypothesis that female peahens will choose to mate with a male peacock that has longer tail, more eye spots over the male with the shorter and fewer eye spots. So here, what are you looking at? Which factor is considered to be dependent? If you look at this, you see that they're saying that mate with male peacock that has a longer tail, more and more eye spots, meaning that if you have longer and more eye spot, you are likely to get more mates. You understand? So the number of mates you get will depend on how many of this do you have and how long your tail is. So the length and the number of the eye spots is independent, and the number of mates depends on how long do you have it and how many are they. Therefore, here the answer becomes Yes, the number of spots. No, the number of mating attempts. How many attempts do you gonna get will depend on how long do you have. So this one is dependent. It depends. The number of mating attempts will depend on yes, will depend on how many females will come, and when they come, they will be having how many attempts you're gonna be having. Actually, the female can come, but because it is interested in you, if you look at this and this, it looks to be confusing. But how many attempts you're gonna have, you understand? Because here they're saying that to choose the mate with the male peacock. So this female will come depending on how long do you have, but how many attempts do you have. When they come, you're gonna be interested, and then you'll be having more successful or more mates to solve that that issue. So the answer becomes B. It looks to be the most correct. This is a karyotype. So in the karyotype, what are they talking about? Read the following statement about this karyotype. Ne? Then they're saying that something unique on this karyotype. 44 chromosomes. Are they 44 chromosomes? No. If you count them, it's 1, 2, they are not 44. So this is ruled out. Ne? It's ruled out. I'll show you why. This is 45 orosomes. This karyotype has 45 orosomes. This, we're going to put a question mark because there is something we need to look at again. Gonosomes are different. Are they different? Someone can say yes, but here there is nothing. They're saying that karyotype is from a gamete. Yes, to look to be from the gamete because it is only having one chromosome. Okay, lastly, karyotype has Down syndrome. No. If you look at chromosome number 21, the two gonosomes are identical. The two chromosomes are identical. Are they identical? No. Yes, because check this. If it is a female, then they're gonna have chromosomes which are what? Identical. So in this case, the answer, because they're saying 45 orosomes, this individual, yes, this individual is going to have, look, is going to have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Check here. So here there are 2. You see? And then if you count them, you'll see that there are 45 orosomes. So you remove this. There are 45 what? Orosomes. So this marks to be most correct answer. So it means that number 2 is correct and the number 4 is also correct. So to become D. Then I'm saying that which example? Why? Because there is no Y here. There is no Y. Someone is going to ask you why. There is no Y there. The Y is not what? Indicated. They're saying that which example is not source of variation? Here they're saying mutation is correct. Crossing over is also correct. But when does it occur? They're saying prophase 2. Wrong. Random arrangement of chromosome, correct. Random fertilization, correct. So this one becomes wrong. So the most correct answer here, because they're saying not. You see? So the question is coming in negatives. So the answer becomes B. So you get the 2 marks there. They say, let's go to the terminologies. And then we find out how is these terminologies. The type of nucleus that contain one set of chromosome, haploid. Then they're saying differentiation. Differentiated cells that can be able to specialize into specialized tissue. I think those people who tried to attend my class yesterday, and yes, I think you got the marks. I think you got the what? The marks. And I also want to talk about one student who just sent me a message, and that message was so good. Yes, the student sent me a message just trying to thank me for what, unfortunately, the student did not send the name. He said that, I just want to thank you for all the resources you have shared and continue to share. They really mean a lot, sir. More especially for us who cannot afford extra classes. I know that outside there are some people who cannot afford extra classes. So that's why this channel was created, so that we can help you out. I really hope that one day I can touch your life. Thank you. The very same way you have touched mine. I didn't know that outside there I touch some people's life. But thank you very much. I pray that you have responsible citizens, because that is my agenda for this channel. And at this point, I don't see a reason for not getting a distinction in the life science after all your tutors. Thank you very much. Also, please use the distinction material. I really appreciate that. Some people, they send me comments, they are very happy for what we are giving them. And if really there is a challenge you find, let me know, or let us know, so that we can rectify those challenges, or we can try to modify them so that at least they become better for you. Yes. But there is only one challenge. I don't know how it's gonna deal with it. Is my handwriting. My handwriting is not good. I don't know. That's why I like to talk too much than, yes, writing. Okay, this is stem cells. Ne? Then they're saying that a characteristics where large protruding jaws. This is called prognanthus. Nanthus. Yes. They're saying the organism that contain genes inserted from an organisms of a different species. Organisms. How do we call these organisms? We call these ones, it will depend on the marking guideline, but scientifically it is called a transgenic organism. Yes, transgenic organism. Yes, so it will depend, transgenic, ne? Yeah, because of you have transferred one gene to the next gene. So maybe there will be another option for those people who have not written. Yes, the type of inheritance that result in the intermediate phenotype in heterozygous intermediate. When you talk about intermediate, we are talking about incomplete dominancy. Incomplete. If you use the distinction material, yes, I think you are getting it right. Ne? Yes, people are using distinction material, you are getting it right. We call it incomplete dominance. African ape share the most recent common ancestor. It's a chimp. It's a chimpanzee. Ne? Is there any other African ape? Don't say Hominid. Those are not apes. They are still humans, but not the modern humans. Yes. So the answer here is supposed to be chimpanzee. Okay, they're saying that the inheritance disorder result in the absence of pigment in the photoreceptor. This is eye. So the moment you talk about eye, you are talking about color blindness. Yes, we call it color blindness. Then they are saying that all genes in an organism of a species, we call this one a genome. I think people who were discussing with, I was discussing with yesterday, think you are getting it right. Ne? They're saying decrease in genetic variation. What decreases? Decreases genetic variation. This one increases, but cloning decreases. So it is A only. The word only must be there. Site for meiosis in plants. I asked a question yesterday, one of the students that give me the where is meiosis taking place. Then someone saying that ovary. Say, he said testis. Then I said that I didn't say that. Give me where meiosis occurs in animals. I was asking where is meiosis because I know they like this. Anyway, here they are saying site for meiosis in plants. Here it's none. It's not ovule because ovule are being produced as a result of meiosis in plants. And the stigma, no. So the answer is none. And they are saying that the variation in blood, the type of variation in blood type, how do you call that? You're either A or B. You can't be both. You're A, B, or O. You can't be O and the same time A, B. No way. So it's discontinuous. So the answer becomes discontinuous variation. And I gave you a clue that if you find out that the variation or the characteristic you are born with are not changing, you still have them, then we call that type of variation discontinuous variation because it's not changing. But the size, look at the size. The way you are born is not the same size the way you are. No. So it becomes continuous variation. Let's go to the next question. They're saying that give the letter and the name of the part. Letter and name of part. That they want two of them, this and this. Forward position is in forward position, and the head and the body are vertical. Yes, it is D. So its answer is D. Is it foramen magnum? Yes, that's what we call. It's foramen magnum. So that's what you're supposed to say. And then you're saying that the functions to support the internal organs. Basically, it's a skeleton. You can say a skeletal system. This, it could be this. Yes, skeletal system. Or maybe, in this case, because they're saying support the internal organs. So basically, is this vertebra. Yes, this tries to support lumbar. You call it lumbar, or lumbar, if it is this, because basically here what is being pointed on is lumbar. Yes, so you can call it lumbar, or basically is a skeletal system, or it is the specific name there based on this is the lumbar. Yes. They're saying that state the shape of the spine in gorilla. Gorilla is the C shape, and then humans is S shape. I think the people who are dealing with the distinction material, you are understanding what I'm trying to say. Yes, I think you are getting it right straight away. Ne? Because why? Because we have been talking about this over and over and over again. Okay. Okay, it was opening the distinction material. Sorry. Yes, let me show you what they are trying to show, to ask you here. Let me make it smaller. Yeah, this is what they're asking you. The shape, you see, is C shape, while this one is S shape. Then they're saying that name the model of location or locomotion the spine has. Check. You see that here we say it's quadrupedalism. Yes, for this, and then this is bipedalism. So those are the answers for that. So it's quadrupedalism here, quadrupedalism, and then this is bipedalism. So yeah, I repeat before you go to your final exams, please use the distinction material. You'll never regret. You will never regret. Yes. And then they're saying that cats inherit many genes that affect the appearance of their coat. One gene control whether the coat is striped or solid color. And this animal, or this cat, the allele, this is A, is for stripe, and then for solid is this, meaning that this is dominant over this. The second gene is meaning that now the moment you talk about the second gene, then automatically this is the hybrid cross. You have, yes, long hair, short hair. Long hair is capital R, short hair is small r. So they're saying that the male who is heterozygous for both this hair mate with short hair, okay, state the type of genetic cross represented above. We say it is dihybrid. Dihybrid cross. And then they're saying that the genotype of the female cat, they're saying that the genotype of the, here they said that the mate with a male mate with the short and solid. So in this case, we see that striped is dominant over solid, and then long is dominant over short. Therefore, it's gonna be short. So if it is short, we say the small l, and the solid small a. But because they are starting with the, okay, you say it is small a, small l. So it's supposed to be small l. So that is the genotype of that. And they're saying possible gamete produced by the male cat. Don't forget that this is heterozygous for both, because they said heterozygous for both characteristics. Therefore, I'm going to have, let me write it here, both characteristics, meaning that it's going to be capital L. What is it starting? Let me see. Long, okay, capital A. It's the same thing. Capital A, capital, no, heterozygous. They are saying heterozygous. They say the heterozygous, capital A, capital L, small l. This is the genotype of that, but they're looking for the gamete. So this is not the answer. They're looking for the gametes, of which this is going to go with this. I'm going to have capital A, capital L. This is going to go with that. It's capital A, small l. And then this with this, small a, capital L. And then this is this. It's going to be small a, small l. So basically, these are the gametes which are going to be formed. So I think when they're marking, it's going to be 1 mark there and then 1 mark there. Or any 2, you get 2 marks there. So that is this answer. This is the answer for this. Then I say name the type of inheritance shown in the this above. How do we call this? This is basically it's complete, but it is inheritance of multiple genes. Yes, multi-gene inheritance. Yes, it's multiple genes. If you are looking at it there, yes. Then they are saying that what percentage of offspring, yes, this same phenotype as the mother. Now here you have to cross and then find out. It's a long question, eh? Okay, so what are you supposed to do here? You're supposed to do the cross and then find out what is the percentage. But we will find out that, remember, if you put it in the, remember the mother, they are saying as the mother. Remember the mother is small l, small l. So if you put it in the Punnett square, you'll find out that we say that it is we say that is small a, small l, small l, small l. So you can complete it. And then here we say it is capital A, capital L, capital A, small l, and then small a, capital L, small l. So when you look at it, we are looking for something like this. So the moment there is capital L, this, all these are out, because all this, it will have all of it. So it is only this 4. This, and this. So it means that it's going to be 4 out of 16, because the whole thing is 16 times 100. So this is not what they want. They want, which is 25%. Maybe they will just say 25 and a percent. You saying 25 and a percent, they will give you a mark in that regard. So the answer is supposed to be 25%. We go to section B. So 50 marks are gone. Section B looked to be a bit easier. Yeah. Anyway, Let's look at it. The diagram below shows the DNA and RNA that is involved in transcription. So if it is involved in transcription, let's look at the questions. What is one? Where is one? One is the strand. This strand is DNA, you see. So definitely this is gonna be adenine. And there are two. Where is two? This one, don't say ribose sugar. Don't say sugar. No, it's deoxyribose sugar. You see why? Because it is a DNA. Yes, because of the thymine. And then they're saying that number three is phosphate group. Phosphate group. Identify the strand that represents messenger RNA. So it's this strand X. Strand X. Give one visible reason, because it has uracil. You see, it has uracil. And then they're saying that describe the process of transcription. This one you have to write. Everyone knows the process of transcription. Even if I don't repeat it, it's the same thing. DNA double helix unwind, weak hydrogen bond break. Then unzip is the same thing, weak hydrogen bond break. To form two templates, one acting as to form two strands, two separate strands, one acting as a template for formation of messenger RNA using free-floating nucleotides, using free-floating messenger RNA nucleotides from the nucleoplasm. And then whereby adenine goes with thymine, adenine goes with the uracil, and then guanine goes with cytosine. Messenger RNA is formed with the coded information which moves out of the nucleus via the nuclear pore to the cytoplasm on the ribosome. Basically, that's what you need to describe. All those points, at least you'll be able to get 5 marks. Ne? As long as you arrange them in order, and then those points are there. There are more than 5. That's why I don't usually talk about, I'm not going to write that much. Explain one reason why transcription is important. It's basically important because transcription results in the formation of messenger RNA, which is used to form the proteins. Basically, the messenger RNA has the coded information for the protein. That's what you are supposed to write. Formation of messenger RNA, which contains the coded information for formation of the protein. Yes. So, then we go to the next question. Next question is Mr. Mega Buck. Hey, I like the name. The wealthiest man in the world. Yeah. Hey. Recently died, and according to this will, Mr. According to his will, he said what? What did he say? The children will inherit. His children will inherit, yes, his wealth or fortune. Since his death, 3 ladies claimed to, yeah, obviously, if there is money, even those people you have never seen, their mothers, the mothers might come and say, no way, this is my son's father. Okay, before this died, his lawyer insisted to do the DNA testing. And then what happened? The DNA test was done, and was done between him or among the wife, the children, and him. All right, so this is his DNA. So the first thing you need to do, when there is 3 people which are involved, the DNA of the mother, and the DNA of the husband, and the DNA of the kids. Yes, if the kid is the son of this parent, of one of these people, basically all the bars, yes, their profile must be matching with this. If here I have 25%, the remaining 75% must match, meaning that there must be no bar which is left out to confirm that that is the parent. Therefore, let's go. Give a scientific reason why lawyer insisted on DNA profile from the woman and their children to determine the parent. Why? Because DNA is unique. Yes, there is no way how you're gonna manipulate it is unique, so that you identify the actual parent of these children, or to identify the actual children of this gentleman. You understand? Yes. They're saying that which child will inherit this is wealth. So you have to first identify. Okay, now let's go. Start with this. This doesn't match. That is just as doesn't match with the mother and the father, then that is out. Okay, we go to this, out. This matches with the mother. So this matches with the mother. Nothing matches with the mother. Yes. So this one is out. This is completely out. What about this? This doesn't match with the mother, but matches with the father. So this is first. This doesn't match with the father, but matches with the mother. Okay? This matches with the mother, but not the father. Correct? This matches with the father. You see? Does it? Yes. It matches with the father. Correct? This matches with the mother. Correct? This matches. Are you sure? Yes, with the father. Yes, correct. This matches with the mother. Correct. This matches with the mother. Correct. This matches with what? With the father. You see that all of them have been utilized. If there is one which is out, that's not your father at all. Then this one, we come here. This matches the mother. Matches the mother. Nothing. It's out. The moment it's out, forget about the rest. You see? So basically, this child, wise child, is going to inherit. Actually, is the son. Yes. Explain your answer using the evidence above in the diagram above. Why is this one a child? Is because all the bands of the child match. All the bands the bands of the child, yes, some match with the mother, the woman, and the remaining one match with the, you see, those which match with the mother is fine. The remaining one must match with the father, or some match with the father and the remaining one will match with the mother, meaning that this is the mother and this is the father. Yes. Oh, if you calculate, if you find it's 50% matching with the mother, correct, and 50% they must match with the father to make it 100. Name one simple source that could have been used to get DNA. It could be blood, it could be hair, it could be saliva, it could be skin, it could be anything which is easy without affecting the mother or the father. Just in case electricity goes, maybe we can be stagnant for some few minutes so that we switch to the system. Anyway, let's continue. They're saying that what is A? This is the chromosome. I told you that when you get this, you have to first, and then this is the spindle fiber. Yes. Then they're saying name the process that is represented. Look, this is single chromosome, meaning that must be meiosis 2. So because it is in the middle, therefore is metaphase 2. Metaphase 2. They say name the chromosome that were represented. How number of chromosomes presented in the parent cell, because there are 2 here, so there were 4. You see, one daughter cell at the end, there are 2. And they are saying give the name of the structure. A, we said is chromosome. And then give the function of structure B, to hold the chromosome in what? In a position, yeah? Yes, you see. And also they are used to pull the chromosomes to the opposite pole. Yes, they contract and then they pull the chromosome to opposite poles. Yes. Describe the process of crossing over. I think people okay, anyway, let me not talk about it. Crossing over, basically you look at chromosomes of homologous chromosomes. The homologous chromosomes, they align together. Chromatids of homologous chromosome, they cross over at a point called chiasma, you see. And even they didn't talk about the phase, so you mentioning the phase is also another mark. The genetic material is exchanged, and then after that they split, and then they will have genetically different genes. Yes. So chromosomes of which are homologous, they align together. Chromatids of homologous chromosome, they cross over at a point called chiasma, and then after that, what happens? They exchange their genetic material, and then they separate or they split to form two genetically different chromosomes. This process occurs in prophase 1. Yes, you can also talk about that, so that maybe if the mark is there. All right, they're saying that draw the phase, the diagram of the phase that occurs after the phase. So here they are looking at anaphase, but when you're drawing, you make sure that, let me show you, make sure that when you're drawing, is actually supposed to be like this. Let me show you. It's supposed to be like this, ne? I don't know whether you can see. It's supposed to be like that. You see, it's gonna split. When it splits, so make sure that these variations are correct. Yeah, so make sure that the variations are correct. So this one is gonna move this side, and then this one is gonna move. So meaning this and this is gonna be black. When you are shading it, you must shade it black, you see. And then this one and this is gonna be it's gonna have a black thing there, and then this is gonna have, you see, you're drawing what will happen. It's a black and a white, black and a white, and then this side is gonna be what? A white, based on what it's shown there. And then spindles, and then you write the phase there, the title of the what? Of the diagram. So meaning that you're right, and then you say that because the question they're saying that the next phase, the diagram of a cell and what do they say is the question? Give a function. Okay, describe the process. We done. Draw a phase. Okay, in there. Yes, diagram of a cell showing anaphase 2. Yes, so basically that's what you're supposed to say, caption, and then you draw these chromosomes I've shown you with, you draw them nicely, ne? The way I've tried to show you, and the variation, because they might be marking the variation, but what I want to show you is that they are supposed to be two chromatids on each side and then separated, and then the caption, so that when they mark you, they give you correct answers, they give you ticks. Let's go to question 2.4. They're saying that familial this syndrome, whatever, is inherited condition caused by the dominant allele. People with this condition have a high level of cholesterol, fats too much in their blood, and then this increases the risk of their blockage, meaning that you are likely to suffer from stroke. The diagram below shows inheritance of the parents of several. Something is important here. Generation. Okay, this is caused by a dominant allele. Look, the person with the, because they are saying that caused by a dominant allele. So that's a point. As I was explaining yesterday, that this dominant allele, you won't miss it, because they know that these days you fail it. So in that case, you come and then you explain. Let's read and find out. Let's read this question. Something is not right here. Is it me who is colorblind? I don't know. Okay, we shall find out. How many generation. You know why I'm saying is because I'm seeing a key here. They're almost the same thing. But if I'm not forgotten, I've not forgotten this was shaded. This was also shaded. It's supposed to be shaded. One shaded. But these ones, some of them are supposed to be shaded. Some of them are not supposed to be shaded. But I remember this was shaded. I think even one of it was shaded here. But it's okay. You'll check on your paper and then find out. Ne? How many generation in this? We count the generation. 1, 2, 3, 4. So 4 generation. Someone is gonna say, no, not this one you don't count. Why not counting the parents? Are they in the same generation with you? Are you in the same generation with your parent? They'll say that our generation need to be like this. Okay. Determine the number of males in the diagram above who are homozygous recessive. So in this case, I'm not going to give the answer for this for now, not because I don't know. Yeah, it's because they shaded. I would have been with the original copy, ne? And I don't have the original copy, ne? Just use the photocopied one. Okay, I would so count the box. You're supposed to count. Let me just give you a clue. You're supposed to count the box. Yes, the box who are shaded. No. Just a moment. Let me just go get just 2 minutes. 1 minute. Get the original paper. Yeah, I'm back. I've got the paper, the original paper. Let me just find out. Let me just shade it for you so that we are on the same. Sorry, I'm not having a memorandum here. Just creating the memorandum as I'm discussing here with you. Yes. Yeah. So it's like I'm also writing the paper. I'm also writing the paper. All right. Okay, yeah, now I've got it. Yeah, this one is shaded. This one is shaded. Yes. This one is shaded. Yes. This one is shaded. Yes. Cool. J is shaded. Yes. Shaded. N is shaded. O is shaded. P is shaded. Yeah, if I don't shade all of them, then you'll find out. Yeah, but let me just show you. All right, they're saying that the shaded ones are the one who are with the problem, and then the unshaded are the one without the problem, meaning that the shaded ones, they are supposed to be either capital T, or capital T, small t. Those are the shaded ones. The unshaded ones are supposed to be, don't forget that this is caused by a dominant allele. If it was caused by a recessive allele, then this would have been the shaded and this is unshaded. But because it's caused by a dominant allele, the shaded ones are the one which have the what? Yes. So now let's go through this. The question is saying that determine the number of males. They are looking for males. Oh yeah, it's the males. So the males are the one with the box. They're saying the males with the problem. So it's 1, it's 2, it's 3, it's 4, it's 5. Based on what I've shaded, yeah, it's 5. 1, 2, 3, 4, 5. Yes, the answer is supposed to be 5. Determine the number of males in the diagram above who are homozygous. Okay, we are done with AA. This answer is 4. Just down here, it's 5. Here I said how many generations? There are 4. Saying, hey, G and are planning to have the 4th child. What is the cross to show? Use the genetic cross to show the percentage probability of child without, meaning that it's supposed to be homozygous recessive. So you guys remember they're saying that they are planning They are planning to have another what? Another child. Yes. So okay, I'm gonna say P2 F2. Phenotype. So you're saying that G is a male who is shaded, who is having the disease. Ne? Yes, you put it there. Cross with a female, which is H, without a disease. You also put it there. And then here you say gamete. Sorry, genotype. So if it's a genotype, because this one is having a disease, but this person came from top here. Where is the G? Oh, G is here. It came from top here, of which this one was also sick, meaning that this one is heterozygous. You understand? Yes, it's heterozygous for the what? For the condition. Yes. So why? Because this one is capital R, small r. Sorry, capital T, small t. This is small t, small t. Definitely this one to be having a disease must have a capital T. There's no way how it's going to be capital T, because this one was the already what? Normal. Therefore now the genotype is going to be capital T, small t, and then H is normal. Remember, is a recessive. Yes, I think now gametes will form as a result of meiosis. So meiosis is going to take place. You form the gametes. It's going to be capital T, small t, small t, small t. Then now fertilization is going to take place. This with that, capital T, small t. This is that, capital T, small t. This with this, small t, small t. And then this with that, small t, small t. So since small t, these are offsprings. You see, F2, it must be in the same line. We come here, we give you a mark. Meiosis and fertilization, we give you a mark. The genotype and phenotype from the question still, we give you a mark. Then we give you a mark here. But there must be a compulsory mark here. A compulsory mark here, the question is saying that use the probability. So in this case, the probability is going to be a compulsory mark. So it's going to be 2 of them, they are normal, and 2 of them, they have the condition. So it's going to be out of 4 times 100, because they are looking for the probability. Yes, the probability, or the percentage probability, which is equal to 25%. So that is the answer for that case. So yeah, let me put it back. I think I'm done with that question. Let's go to question 2.5. These papers were too much reading. Anyway, lactose is the sugar in milk. Lactose is found in milk. It's digested by the enzyme called lactase. Then some people cannot produce lactose, and therefore they become lactose intolerant. And then the condition is caused by recessive allele. This means that the people with the homozygous recessive, they become lactose intolerant. People from different countries, okay, were tested to determine if the lactose intolerant, they were lactose intolerant. They are saying that the table below shows the number of people tested in each country and the lactose intolerance percentages. Okay, let's find out. The population. Here you are having the percentage and then tested people. The population size of country B. Where is country B? Okay, is 190 million. They're saying that calculate the number of people in this country likely to be lactose intolerant. Here they're saying the percentage of people who are lactose intolerant. So here these are the number of people they tested, and this is the percentage of the people. You understand? Who are lactose intolerant. So meaning that in this case, because this is the percentage, so I'm gonna say 12 divided by 100. That's the meaning of percentage times the 190 million people. So when you get the answer, I think it's around 22.8 million people, something like that. you can calculate and then find out the actual answer. But that's how you're supposed to do the calculation. There is another method, but let's just go with one as long as you get the correct answer. Which country is most likely to be having the least reliability? Check here. Remember when I was discussing yesterday, I said that people, you need to know what is it. So the answer is E. Why? Because we use the same fewer number of people. Remember, if you want to increase the reliability, you increase the sample size, the number of people who are in this investigation. You understand? Yes. So we go to the next question. Yes, we go to the next question. Just a moment. Yes, let's go to the next question. Give a reason for your answer. We said that because you have used this fewer number of people. Provide 2 ways in which the validity of this experiment can be improved. Validity is you are keeping things constant. Yes, use the same number, maybe the age. Remember, age can also affect the people. Same kind of individuals. Yes, you are busy increasing the what? This validity. Keep the same type of the food you are giving them, because they might have different concentrations. Yes, meaning that the concentration of food or the type of food you are giving these people, which has the milk, sugar. Yes. Then there are so many. Describe the planning there a lot, because we are using different countries. It depends on where. If you are going to fly to different countries, then therefore you have to plan in advance, maybe requiring permissions or visa things, the budget. Look at the budget. Ask for permissions for these people. Are we going to be experimented about? You have to look at the suitable time for these individuals. So there is a lot of planning steps you can consider. Then let's go to question number 3. Yes, question number 3. Yeah, we are almost done. That was question number 3. Okay, it's too much of talking than writing. Flies lay eggs in urine and fish that stuck on the wall of the what? Of the sheep. The eggs hatch and then larvae damage the sheep's skin, leading to the infections. To reduce this problem, farmers in New Zealand have been developing sheep with no wool growing on their legs. Then they're saying that the back side, that's the bottom, or the under side of their abdomen. Farmers have crossed different breeds of sheep to produce offsprings with desirable characteristics. And they are saying that the characteristic of other breeds of sheep are shown in the table. Okay, they're saying that use the evidence from the table to describe how the farmer could use selective breeding to develop. So if you look at our distinction material, it shows you what exactly you're supposed to do when you are dealing with the selective breeding. Yes, it's here. So yeah, there is basically what you're supposed to do. In this case, the first thing you're gonna do is to select the individual who have what you want. So basically, you want these legs. You want bare legs, ne? Did they say bare legs here? Yes, the farmer crossed breeds, okay, offsprings with desirable characteristics, and then with other sheep. Okay, they're saying that produce. Yes, he said it here, that use the evidence to describe how farmer could use selective breeding to develop the sheep with bare legs. You see, bare legs. And then backside, bare backside, not abdomen. They're looking for backside. So meaning that this individual is gonna cross this, because this has but they doesn't have the bare backside. Therefore, it's gonna cross the one which has bare legs and then the backside, and then it will deny sex to this. So meaning that this one will not be given sex, only this one will be interbred. And then yes, you keep interbreeding. This is what I'm trying to say. So what happens is this is what you can say. For example, to increase the quality of is here. Suppose you want to a variety of cow to produce a lot of milk. Now in this case, suppose you want to have this sheep with these characteristics they are looking at. You see, now you are writing. Choose the cow in the herd to produce that can produce most milk. Choose the sheep that is this, the one which can have bare legs, and also so these two, because they asked you how did use the evidence describe how the farmer could use. So we did select this and this because they have these desirable characteristics. So after that, then allow these offsprings. Let only allow these cows to reproduce. These ones which have the desirable characteristics, these sheep, you allow them to interbreed. That's why I'm saying that you are denying this one sex. And then after that, what happens? The one when they get the offsprings, yes, the offsprings that produce these desirable characteristics, the one who has bare legs, and then this, you cross them again. You see, you cross the offsprings that can produce this characteristic. You let this offspring reproduce again. You understand? So at the end of the day, you have animals with this and this together. One animal with those two characteristics. That's how you're supposed to explain. Then give two tabulate. Here you're supposed to draw a table. I told you guys, you draw a table, complete table. You don't draw a T table. This is wrong. You get a 0. So don't cry when you find a 0 there. So tabulate the difference between selective breeding and natural selection. Selective breeding, basically, this one occurs naturally. So the nature will select. Here it is human needs. Yes, then basically it's like artificial selection versus natural selection. Environment will do the selecting. Human will, is the selective force. You understand? Yes. It occurs within a specific species. This one can occur in multiple species. Multiple species, yes. Because you are just crossing, yes. So you only select what is best, what you think it is good for you. So basically this is the differences you're supposed to write about. Explain one disadvantage of selecting breeding of sheep in this question above. Basically, when you do the selective breeding, this sheep might lose important characteristic which might help it to be able to survive in the wild. You see? Yes. Maybe the hair, and then you don't want this hair, and the hair is there to protect this sheep so that it can sustain in different temperature conditions. And then when you just want to wipe it out because it is doing the caca on the wool, because you don't want it. So at the end of the day, that character might get extinct. So that's whenever you're doing selective breeding, you are not leaving the nature to select against those who are not best suit. Then you are doing some disadvantages to the animals. Yes. Then when we go to question 3.2, the diagram below shows the modern swordfish. The ancestor of had short sword that that yes, the modern swordfish have long sword. Yes, the modern swordfish have long swords. Yes, the swordfish use their swords to kill the prey, meaning that they're using it to kill the prey too much. The injured prey is easier to eat. You see, it's easier to eat and is easier to catch and eat. Something is important here. Okay, they are saying that the diagram below shows one theory of how the length of the sword it got evolved. Here is many generation saying the sword grew longer each time the swordfish uses its what sword more and more. Oh, the more you use the structure, the more it becomes bigger and stronger. So it means that that's Lamarck's theory. Oh, they're asking you a question. Suggest which scientist suggested the theory above. That's Lamarck. Explain one reason why this theory is rejected by most of the scientists. Because organisms don't evolve because they want to evolve. This sword did not evolve because the fish wanted it to evolve. And then there is not enough evidence to show that this acquired characteristic during lifetime is passed to the next generation. You understand? Yes. Describe how natural selection could result in modern sword. So here you're using Darwin's theory. So what are you going to write? So here you talk about the variation, that there is variation among the swordfish. Then you tell us the variation. Some swordfish had long snout. How did they call it? They call it sword. The sword, yes. Some swordfish had long swords and some swordfish had short sword. Yes. Those with the long swords, here they said it. They say that they could. Yes, check here. Those with the long sword, yes. What happened here? The one with the long sword, what happened? Yes, they are able to kill the prey and catch it and also eat it. Therefore, they survived in large numbers. While those with the short sword, they couldn't get or catch the prey and eat. Therefore, they died in large numbers. Those with the long sword, they passed their genes. This is called natural selection. Those with the long swords, they passed their genes to the next generation of the long sword. After many generations, all swordfish had long sword. See? Yes. So when you're explaining it, you're supposed to link it to this example. Yes. Then the next question they're saying that describe. Yes. So we are done with that. So variation. Some swordfish had short sword and then long sword, and then those with long sword could catch the prey and kill it, and then they survived in large numbers, while those on a short sword, they couldn't, and then they died in large numbers. Those with the long sword, they passed their genes. This is called natural selection. Those with the long swords, they pass their genes to the next generation. Yes, after many generations, all the swordfish had long sword. So that's what you're supposed to explain in that regard. Then when you go to 3.3, 3.3, they're saying that the table below shows the average cranial capacity in hominids fossils. Okay. State one Australopithecus africanus species you have learned about and give. Yes. So what are you going to give? And give the common name, and then so basically you just supposed to choose maybe Australopithecus sediba, that's Carabo, who discovered it. So all those things, they're saying Australopithecus africanus. They're saying did they say africanus? Australopithecus. It is wide. Afarensis. So it's only Adipithecus. So Australopithecus Lucy Johansen. Then Australopithecus africanus, you can, it says place Robert Broom. Yeah, Littlefoot, Clark, and then Australopithecus sediba, Carabo, Idibega. Then all this, you can choose whatever you want to choose, and then just tell us Australopithecus, then what is it, and then you tell us who discovered it. So you choose any of these Australopithecus from here up to there. So you choose any. Yes. And I was emphasizing how do you memorize that. Step 2, advantage of having a large cranium. Large cranium is going to bring about more brain cells. And then these more brain cells are going to link, is going to bring about intelligence. And then if you have more intelligence, meaning that you can think more, you can reason more, you can have more skills. All those are some of the advantages of having a large cranium over a small cranium. Then that's what you're supposed to write. Then you're saying draw a bar graph. Draw a bar graph to compare the average cranial capacity of Homo. Guys, this question was a little bit not supposed to be rushed on. You know why? Let me show you. They're saying Homo species. Some people are busy touching on their head. Oh, I drew everything. That's why I said that don't rush. Don't finish the paper in 1 hour and then you say, yeah, I'm done. You think that the examiner is a fool. No. Okay, they're saying Homo species. So it means that your bar graph was supposed to be only those individuals, because those are the Homo species. You see? So from there, after drawing that, then what are you supposed to do? You come here and then you tell us, you draw. You're supposed to draw the bar graph, but now what goes on x-axis, what goes on y-axis? Hominids, they go on the x-axis. So you tell us here the hominids, and then the y-axis, they're saying number of fossils, they're saying cranial capacity, because they're saying cranial capacity. So meaning that the cranial capacity is supposed to be here. Did you give the caption? Did you give what is here? You put the names, the spaces are correct, the scale is supposed to be the same, depends on the scale used. Yes, the small one is, the smallest is 640, the biggest is 1350. Maybe you can start from 0, 500,000, something like that, or you can be 250. Yeah, 0 to 50, 500 to 550, 600, 750, something like that. Yeah, so like that, 0 to 50, then 500, 750, 1000, 1250, 1.5. So like that, it must have the same range, so that we come here, we say caption, we give a mark. We say correct type of the graph, we give a mark that it's a bar graph. Yes, it doesn't matter. And then caption, you put the caption, you give a mark. The spaces are correct, it must be correct, same spaces, same size. And then scale, we see the scale on x and axis. The scale, we give a mark. Have you labeled it correctly? X and y, you have labeled it correctly, give a mark. And then plotting, maybe we can give you 2 marks there, 1, 2, 3, 4, 5, 6. So maybe that's how we're gonna be marking it, so that now you get the 5 marks, all of it. Then they're saying that explain the genetic evidence to support the out of Africa hypothesis. I was explaining it even yesterday, and then I was giving an example with these kids, and then people. So this is what we are supposed to say, that inheritance of only inherited only from paternal line. Analysis of mutation of this mitochondrial DNA show that the oldest female, yes, the oldest female, yes, the oldest female, what happened to it? The oldest female ancestor. Just a moment. Thank you for, yes, I was explaining that the mitochondrial DNA. So here, if you look at this question, check how the question is saying that explaining how genetic evidence support the hypothesis that ancestor of Chinese Homo erectus fossil originated in Africa. So here you are showing that this mitochondrial genetic evidence, and we have mitochondrial DNA. It is inherited only from the what? The maternal line. You understand? And then the analysis of mutation of this mitochondrial DNA show that the oldest female ancestor with the were located in Africa, and all look, and all now look, and all these Chinese, and all these this came from what? From what? From Africa. So basically, that's what they are looking for in that regard. Yes. So the next question is saying that study the phylogenetic tree. I was clear yesterday when I was trying to explain this. Study the phylogenetic tree showing the evolution relationship between the birds. These are the birds, the eaves and the crocodiles. Ne? Answer okay. The following questions. When did this branch, when did the Eve or the birds or branch crocodile, when did they branch from each other? So it will depend on this will depend on, if you look at this, I don't know whether I see it properly. I don't have the original again. If this is 1 year, 2, 3, 4, 5, 6, from here to here is 1 year, 2 years, 3 years, 4 years, 5 years. Basically, they separated here. This is when they separated. If it is in terms of years, they separated in million years, meaning that it's 5 million years. But this is where they separated from. I don't know whether they forgot to put the numbers here, 0, 2, 4, or depends on what we're supposed to be here. So they will see how they're gonna mark it out. But it was supposed to have a mark there. Yes, it was supposed to have figures there. Let me check from the original paper. Then the original paper has nothing. Okay, it has nothing. So I think they will put a concession there. But if it is regarded as 1, then we would have said that then it would have been 0, 1, 2, 3, 4, 5, meaning that they separated 5 million years, meaning that is 5 million years ago. That's when they separated. But they're saying that give the term that describe the structure shown in the symbol of the forelimb that was inherited in common ancestor. Which structure? Oh, this one. Yes, forelimb, this one. How do we call this? How do we call this? We call it pentadactyl limb. Let me show you where I put it in the booklet here. The distinction material. Yes, it's here. We call it pentadactyl limbs. You see? It's here. Pentadactyl limb. This one. This is what they are looking for. So pentadactyl limb. And then they are saying that that is the answer for that question. Then the next question, the second last question, they are saying that too much reading. Okay, that imps and ostrich are both large flightlet birds. Imps are only found in Australia continent, and then ostrich are found in Africa. All right, tell us the story. We are listening. Okay, this is the imps, this is ostrich. The original hypothesis about the evolution of the imps and the ostrich was that they share a common ancestor. They share common flightlets. This is a very important thing. Share. Okay. The sea separated the ancestral population during this. So meaning that this is the force which separated the continents. The Australia and Africa, yes, more evidence has resulted in a new hypothesis that the ancestral population of imps and ostrich was highly flying small ancestral birds that could fly long distances and migrate from Central Asia to continents in the southern hemisphere. The new genetic analyses show that mutations in DNA cause modern birds to lose the ability to fly eventually. And then describe the question: how the high flying small ancestral birds evolved into different species of large flightless birds, ostrich and imps, on the other continents. So how are you going to explain this? So basically here this is speciation. Yes, and this kind of speciation is the allopatric speciation because there is a geographical barrier which is separating them. So what are you going to say here in the so you can say that at first there was one big mass with a common ancestor of flightless birds, because they said it the originals, they're saying that we're sharing a common, common flightless bird, and then separated into this by this. So meaning that at one time there was one large mass with a common ancestor with a common flightless ancestor. You understand? Yes. Or this flightless ancestor lived on one big mass at the beginning. And the mass was divided into Australia and Africa, whatever. Yes, due to continental drift. I see. Yes. And then these organisms which were in Africa, where exactly? Which organisms were in Africa and Asia, they could not interbreed because they experienced different environmental conditions, and because of the no gene flow, the natural selection occurred independently. Therefore, they became genotypically and phenotypically different. If you remove this water mass, they say the sea. They said it somewhere. Yes. These birds cannot, interbreed when they mix. They cannot interbreed to produce a fertile offspring. Therefore, resulted into large flightless birds, ostrich and imps. Yes. So this must be written at the end, because this might be a compulsory mark, because this is what you are looking at. And also a common ancestor of flightlet birds could be a compulsory mark, meaning that if this question is 7 marks, you find out that 5 marks is for free, meaning that you can score it anywhere. But you saying a common ancestor is a marking point which is compulsory, and also you saying that mentioning these birds, yes, because that's what we are looking for, might be a what? It might be a what? A compulsory mark. Yes. So if you're a teacher and you are here, please try to look at it on the memorandum and then find out where to put a mark, not just giving marks. Yes. So that's how you explain that. Then the last question, but not the least, O is the last one. Identify. Here you have. Yesterday I was at one of school. I was in 3 schools, né? But there is another school where my colleague called me. It's called Wiseman. All right, yeah. As I was about to leave, a student asked me the same question about punctuated equilibrium. How do we differentiate between punctuated equilibrium and gradualism? I don't know why this always disturbs students when it comes to explain, but let me explain it. Here it is punctuated equilibrium. The diagram you see here is punctuated equilibrium. It has periods of rapid change and long periods of no change. Short period of rapid change and then no periods of change. You see, short period of rapid change from here to there, and no period from here. No periods of what? Change. From here, you see, rapid change. Rapidly they change without seconds and seconds. You understand? And we say that this is caused by a mutation. Imagine from here, from this kind of rat to white, immediately in the morning you find yourself that. And then from this kind of rat to a dark one, from a gray to a dark one. We don't have, it's caused by a mutation. So that's number one. Punctuated equilibrium is caused by mutation. Number two, it has no intermediate at all. Because how is this linked to this? Nothing. It's just rapid change. Then number three, it takes place within a short period of time. Short period of time. If you compare it with gradualism, which moves slowly, you'll find out it's caused by natural selection. The nature must select, and the nature, it's slow. Select small. Then, when you talk about it has intermediate. From here to there is an intermediate, you see them. And then it takes long period of time. So now, if you come here, identify the concept of evolution illustrated above. So here it is punctuated equilibrium. And then give the definition of the concept evolution as shown in the diagram above. Basically, this is evolution with rapid periods of change and long period of short period of rapid change and then long periods of no change. Yes. So this also is a type of speciation. It's trying to show you how speciation occurred. So guys, I think that was the paper. How was the paper? How was the paper? Ne? Yeah, I think the paper was not that bad, but it had too much of reading. Even paper one had too much of reading, and we know that our kids, they don't like to read too much. They are not straightforward like other provinces I've seen. Yes. So if I look at the analysis of these papers they have written in terms of life science, paper one, it was not that difficult, but was too much words. Was too much words. Paper two, the same story. It's not too much. Our students don't like to read. Ne? And I know you are there, you know that you don't like to read. Keep reading. It's not, you don't like it. Ne? So you guys, thank you for being here. Don't forget to get a distinction material book from the description below. Tell us what you need us to talk about. If you have any challenge, let us know so that we can help you out. And then also, don't forget to tell your teachers to help you out. We are always here to help you out, you guys, so that at least you have a better life. Yes, so that when I get to my retirement age, at least I know that I helped maybe 1 billion people. Yeah, 1 billion people. People maybe in South Africa, at least 50% of them, at least they pass through my hands or somewhere. I helped them a bit. At least I will be in my retirement with happiness. Anyway, I wish you the best in your finals. I'll be here still to help you. I've not stopped from helping you. I'll help you even if you have done writing your paper.